Hello students, today we are going to study a very important topic, that is principles of cancer. In today's session, we are going to understand the definition of cancer, the types of cancer, the properties of cancer, and the pathogenesis of this disease. Before we understand the definition of cancer, we must understand the process in which a normal cell functions. When we talk about how a cell divides, we also talk about cell cycle, which includes mitosis and meiosis. The mitotic phase or M phase is followed by G1 or interphase, which is followed by S phase, in which growth and DNA synthesis takes place, and finally the G2 phase, in which final preparation for cell divisions take place. In addition to this, there is also a G0 phase. A cell in G0 phase will not undergo any more division. Now, this process of cell division in a normal cell is regulated by two important processes. So, you have the process of proliferation, which is responsible for growth, and the process of apoptosis, which is nothing but the programmed cell death, which helps to control the growth of the cells. So, there is always a limit on the cell growth, or there is always, what you can say, control on how much growth is there of a particular cell. However, when this balance disrupts between the process of proliferation and apoptosis, what we get is uncontrolled cell growth. So, cancer can thus be defined as the uncontrolled growth and spread of cells. This is a representative image that you can see in this slide to show you how cancer cells show uncontrolled growth and proliferate through the normal tissues. So basically, the term cancer is not a single disease, but it is a group of diseases which is caused when abnormal cells divide rapidly and spread to other tissues and organs. Why exactly is cancer called a group of disorders? Because cancer can be of different types. Depending on the type, the properties may vary. Though uncontrolled growth is seen in all metastatic cancers. The cancer could be of different types based on cells of origin, from where it is originating, the organ that is affected, and whether the tumor is localized or malignant. Let us understand the first type of cancer based on the cells of origin. The first type that we are going to study in this classification is carcinoma. Now, carcinoma are the cancers of epithelial cell. One example here is basal cell carcinoma. The second type of cancer is a cancer of the tumor or the tumor of connective tissue. An example is osteosarcoma. The third type is leukemia. So these are basically the cancers of blood forming cells. Example is chronic lymphoblastic leukemia. The next type is lymphoma. Now these are the cancers of the lymphatic system. This could include tonsils, the spleen and thymus. So this could get affected. One example is Burkitt's lymphoma, which is an example of the tumor or cancer of the lymphatic system. Next is myeloma. Now myeloma are basically the cancers that originate in the plasma cell that circulate in the blood or form part of the blood. Example is multiple myeloma. So these are the types of cancer which are based on the cells from which they are originated. The next type of cancer is based on the type of organ that is affected. For example, the cancer that affects the lung is called lung cancer. The one that affects the breast is called breast cancer. Similarly, liver cancer, the one that affects the liver, brain cancer, the one affecting the brain, thyroid cancer, stomach cancer, and so on, depending on the organ that is. The next type of cancer is based on the degree of proliferation. Now, cancer can be of two types, that is, they can be benign and malignant. Benign means they are confined to a particular location. They do not spread, they do not invade other tissues. Whereas in case of malignant, they are capable of invading surrounding tissues. So they are capable of spreading. They are the most lethal type of cancer. So far we have seen the definition of cancer, the different types of cancer we have seen. So now we will expand our knowledge to understand the properties of a cancer cell and the pathogenesis of this particular disease. Now let us understand the properties of cancer cell. Now cancer cell, they will show abnormal proliferation, differentiation, and you will see that the process of apoptosis is suppressed. So they will show an increased lifespan or survival as compared to a normal tissue. So now when we are going to understand the properties of cancer cell, we are going to understand that relative to what we see in a normal cell. Now when we understand the properties of cancer cell, it is important for us to understand the difference that exists between a normal cell and a cancer cell. So the first important difference is that when you talk about a normal cell, they have density dependent cell proliferation. Now what do you mean by density dependent cell proliferation? For example, if you have a cut on your hand, in that case there will be growth of new cells which would help in healing of that wound. Now this process of growth of cells would be limited till the time the healing is complete. After that, these cells won't continue to grow or will not continue to form a lump at that place. So in a normal cell, there is always a limit as to how much the cell would grow. That means the growth is arrested once they reach a finite cell density. This is not seen when you talk about a cancer cell. They do not show density dependent cell proliferation. They continue to grow, and so you would see a tumor formation or a lump formation which would take place. So another important difference in addition to this, what you will observe is when you talk about any cell to grow, it requires something called as a growth factor. Now this growth factor requirement for every cell is defined. It requires it in some optimum amount. But when you talk about cancer cell, this requirement is reduced, which means that with very little availability of growth factor also, it can grow and proliferate. So because of that, it has its ability to proliferate or grow much more as compared to that of a normal cell. Next important difference that we will observe is when you talk about a normal cell, they have presence of something called as E-cadherins on their surface. Now what is this E-cadherin? So these are a special type of proteins which give adhesive properties to cell, and because of these adhesive properties, the cells will be able to maintain some restraint. They are unable to invade other cells. However, when you talk about cancer cells, they lack these adhesive proteins. So because of that, they are unrestrained in their interaction and they have the ability to evade and spread to other tissues. These cancer cells also have the ability to release something called as protease, which is a protein digesting enzyme. So this enzyme digests the extracellular matrix of other tissues, and because of that, its ability to evade or ability to metastasize is much more as compared to if you compare that to a normal tissue. Another important feature is that normal cells have a defined capacity of angiogenesis. Now angiogenesis is the formation of blood vessels. Now they have a defined capacity in a sense they can grow and differentiate normally, and also they can undergo apoptosis, which is something called as a programmed cell death. That is, after a certain span of a number of days, they undergo programmed cell death, and so they have a certain defined lifespan. Beyond that lifespan, they will not survive. This is not seen in case of a cancer cell. So what we observe is that they have increased ability of angiogenesis. More number of blood vessels they can form, and because of that, they can undergo abnormal differentiation, and they fail to undergo apoptosis. Programmed cell death is not seen, and they show increased lifespan. Now let us have a look at how exactly this cancer cell differs from the normal cell. So I said that they undergo differentiation, proliferate and grow very rapidly. The way in which they grow is not controlled. So you can see over here that there is variation in the size and shape of the cancer cells if you have to compare it with a normal cell. Also, their nucleus is much more larger as compared to that of a normal cell. They have abnormal number of chromosomes and they form clusters without a boundary. So they are poorly differentiated. This is one of the very important distinguishing features which separates them from the normal cell. That is, even though they undergo differentiation, the differentiation is not normal. They are poorly differentiated cells. Now, to understand the process of formation of cancer, we need to remember some important players in this process. So remember that there is something called as an initiator, which initiates the process of mutation. Okay, and there is something called as a promoter, which promotes the proliferation of this mutated cells. So remember one thing, or two things rather, that there is something called as an initiator, which is responsible for causing mutations. And there is something called as promoter, which promotes the growth of this mutated. Now, what exactly is this initiator? So, initiators basically are agents that induce mutations or they transform a cell. So, initiation is going to be the primary step. Primary step because unless that mutation is induced, the cell will not show different properties than that of a normal cell. So, these initiators are also called as carcinogens. They are the ones that induce mutation. Let us understand the different types of carcinogens. Now, they could be chemical carcinogens, for example, benzene. They could be physical, for example, ultraviolet radiation. It could be infectious pathogen, example is Epstein-Barr virus or even hepatitis B virus. So now these carcinogens, they tend to damage or destroy some genes, and by this process they induce mutation. Now these are some examples of carcinogens which I have taken. There are many more apart from this, but you just have to remember certain number so that you know what is the basic classification and what are some of the examples of this carcinogens. Now once this particular cell which is exposed to the initiator or carcinogen is altered or undergoes mutation, then in that case it will undergo proliferation over a period of time. Now this particular cell which is exposed to a mutant or which is exposed rather to a carcinogen or an initiator is also called as an initiated cell. Now we have talked about the cells that undergo mutation, that undergo initiation. We have talked about cancer cells and how they differ from a normal cell. But at the cellular level, what type of changes occur, what kind of mutations occur, we need to understand that as well. So with the help of this image, I will try to explain at the cellular level, at the molecular level, what level of changes tend to take place because of which the cell that has undergone mutation differs from a normal cell. So consider this image. In the top I have circled some cells which are the normal cells. Now if you observe this particular cell, you will see that there are a pair of chromosomes which are X chromosomes. So X1 and X2 are the two chromosomes. Okay. So you will see in some cells X1 is active and X2 is inactive. In some other cells it is opposite, that is X2 is active and X1 is inactive. So basically if you consider cells, they are a mosaic of different types of there is basically a mosaic of cells with different types of X chromosomes being active or inactive. So this is how a normal cell structure basically would be. Now if you focus on the cells which are below, the darker ones which I have circled, you will observe that they have the same consistent pattern of X chromosomes. That is, there is one consistent pattern. If you compare these cells which are circled below and the cells which are at the top, you will find that the cells that are at the top, that is the ones which are normal, do not show a consistent pattern. you will see an alternate switch between X1 and X2 being active or inactive. However, over here below you'll see a consistent pattern. Now what inference can you draw from that? This means that this particular cells have undergone some kind of change or have undergone some kind of mutation, or rather they have been derived from a single cell which has undergone some alteration or some mutation, because of which their expression pattern is different from what you see for a normal cell. So these are some example. This is just an example to explain how the expression of this X chromosome differs in case of a cell that is genetically altered. Let us understand now the whole process of how a cancer develops. So there is an initiator which comes into the picture first. Okay, initiators. What are the initiators? What are different types of initiators? We have already seen. Now this initiator will induce mutation. So as you can see at the top in this image, you have number of cells, and the cell that has undergone change or alteration is shown in a different color. Now this cell which is genetically altered or which has undergone mutation will grow or proliferate in the presence of some promoters, which could be in the form of growth factor promoting oncogenes. Now as it grows, it will undergo some additional mutations. Now in this process of undergoing mutations, what will happen is that the cell with these mutations will confer some advantages on this cell. So, like for example, if a cell undergoes mutation which inhibits the process of apoptosis, then in that case the cell will not have a defined lifespan. It will just continue to live for a longer period of time. If there is some kind of a mutation because of which the process of angiogenesis is increased, then in that case the cell will just continue to grow and differentiate uncontrolled. So these are some kind of mutations which will confer advantages on that cell. Now the cells with such type of mutations which confer advantages tend to become more dominant in the process. So over a period of time, the cell which undergoes such kind of selective mutations tend to emerge as more dominant cell, which is also called as clonal selection. Now once what will happen is this dominant cell will continue to grow much more rapidly. So you can see this cell over here which I have shown in pink color. Now this pink color is a mutated cell among this series of mutated cells which are shown in white color, orange color. But this particular pink colored cell is much more dominant cell. It has undergone some mutations which have rendered it more dominant. These mutations has conferred some additional advantages on this cell. So as you can see in the last image, that is the last picture in this particular image, you see that this pink colored cell has grown and proliferated much more rapidly as compared to the other mutated cell. That is, it has become much more dominant, and these cells will now continue to multiply, grow, and what you get, the end product is a series of malignant cells which will now grow and invade other tissues. So this is exactly how a cancer cell evolves. So initially, I repeat, you have initiator which will induce mutation. Now this mutated cell is also called as an initiated cell. This initiated cell will immediately not start growing. In the presence of some promoter or some growth factor promoting oncogenes, this will proliferate or this will grow. Now once it grows, what happens is that as it grows, it is exposed or undergoes a series of mutations. Now these mutations keep on conferring some advantage on that particular cell. So for example, here initially you have this white cell. From this white cell with additional mutation, you have this orange colored cell which is emerging. This orange colored cells undergo some additional mutation and you have this pink colored cell which is emerging. Now among this process, the cell which undergoes the most selective or the kind of mutations which confer advantages on that cell, like for example, the stimulation of angiogenesis, inhibition of apoptosis. So these processes confer some advantages on that cell to continuously grow or proliferate. So that particular cell emerges as the most dominant cell, which is also called as a process of clonal selection. So ultimately, out of clonal selection, a very invasive malignant cell emerges out. This cell has the ability to grow rapidly and invade other healthy tissues. This is basically the most advanced form of cancer cell. Okay. Now let us just understand this whole process with the help of one example, that is how colon cancer develops. So as I told you earlier, there is something called as an initiator, because of which there is a mutation which is induced. So now you can see there is a layer of cell and there is one single cell which is mutated, which is shown in violet color. Now this cell, in the presence of some growth factors, will proliferate. So you have seen that now it has proliferated and it has grown into number of cells. Now this particular cells continue to grow, proliferate. There is clonal selection, and in this clonal selection process, the most cell with the most mutations which are conferring advantages on that cell will continue to grow, become more dominant. So you have the process of growth of cells where initially there is formation of small adenoma, which you can see. Now these cells will just continue to divide and there is formation of intermediate adenoma. This will further result in development of large adenoma, and finally what you will see is that this particular cells, they will metastasize, that is, it will tend to proliferate, invade other tissues, and mainly this process of invasion of other tissues is happening much more rapidly by invasion of blood and lymphatic vessels. So through blood and lymphatic vessels, it will spread to other tissues, and that is how the growth basically takes place. So that's all for now in today's session. The remaining parts, we will see in the next